It's my pleasure to introduce to you Dr. David R. Nelson. He holds a distinguished position of Professor of Medicine, Interim Senior VP of Health Affairs, President of University of Florida Health, and a Director of the Clinical and Translational Science Institute, CTSI, at University of Florida. Dr. Nelson obtained his medical degree at State University of New York, Syracuse, and completed his GI training at University of Florida, where he stayed on his faculty and advanced to the position of professors in the Department of Medicine and the Department of Molecular Genetics, as well as in microbiology. He also holds the distinguished position of Associate Dean of Clinical Research in the College of Medicine. Dr. Nelson's tenure as a director of the CTSI has become a catalytic hub for translational sciences at UF and throughout the country, and has been awarded more than $42 million of awards from NIH. Dr. Nelson has authored more than 200 publications. He has received more than $80 million in research funding and is the leader of the development of national and international guidelines related to management of viral hepatitis, including for the CPIC guidelines for the use of genomic-guided Hep C therapy. He also represents the United States on the World Health Organization Committee for Hep C guidelines. Please join me in giving a warm welcome to Dr. David R. Nelson as he gives an excellent presentation on private-public partnership targeting real-world data and Hep C therapies.
Well, thank you. It's an honor to be here, especially under this occasion. I didn't know Leon Schiff, but I know his son, who, if they're anything like each other, it's a wonderful man, and obviously read his textbooks. I was a fellow 26 years ago and met actually Gene Schiff for the first time, and he kind of took me under his wings and has been a distant mentor, keeping me involved in trials and others, especially when my mentor at the University of Florida had left. So I have a warm spot in my heart for Dr. Schiff, as well as a lot of great collaborations here at the University of Miami. I used to be a translational kind of functional immunologist, and I'm not going to talk about Hep C and the discovery, and you've heard that story before, but over the last 10 years, I've become much more interested in trying to develop infrastructure to help clinicians and patients make better decisions, and really learned about big data and cohorts. And I want to share with you at least the story around hepatitis C, then transition into how we can leverage that same infrastructure here in the state of Florida with some partnerships that are already underway, but really looking at an area where I think all of us are interested in getting better data. So I have some conflicts. I don't take any money from these, but we obviously get research support to help. And we have a fair amount of NIH and PCORI and other funding. So I'm going to start by just talking about what are the regulatory challenges and hurdles, why real-world data is now the hot term. As you probably know, every pharma company in the world now has these massive real-world evidence divisions or departments, and there's a reason for that, which I think we helped contribute to. I'll show you the model that came out of the Hep C world, and I'll show you some examples just to highlight how these observational cohort data sets can actually speed, enhance, and improve the drug development and delivery of care. And then I'll talk a little bit about pragmatic trials and how we've at least leveraged some funding and industry relationships to try to get earlier pragmatic trials into phase 3. And then I'll end just talking about another liver example, but some collaborations we have with Miami and others across the state at putting together a really unique data set for researchers at our institutions to use. So there's a big gap, and this is not going to be news to anyone. So phase 3 trials are designed to succeed and get a drug or device approved. Therefore, they're very narrowly focused, typically around a deliverable outcome, and they're very controlled, and as you know, the patients that enroll in those don't typically look like the average citizens and patients that we take care of. So there's a big gap after a drug or device gets approved because there's a lot of evidence that hasn't been generated. The goal is to get these life-saving drugs to market quickly, which is a good thing, but along the way, there's a lot of unanswered questions around, do these drugs or devices work in patients like me? Patients like me are the ones in the trial. Patients like me that we care about are the Hispanic, rural, inner city type of person sitting in Miami. Those typically are not represented well in these trials, and especially with the social determinants of health issues around delivery care. So there's often a lot of lacking data, and the way the FDA has approached this is they say, okay, company X, get your drug on the market, and now you have to go fill in the rest of the story. And those are called post-marketing approval studies or phase 4 studies. And typically it's looking at older ages, people with more severe diseases, comorbidities, medications that might not have been tested, especially, say, in an HIV community. And those have historically failed. They're typically very late in the game and don't really help fill in the gap. And I'll talk more about that. So I just kind of put on a hypothetical 3 clinical trials, phase 3 trials, and which would have led to the approval of, let's just call it drug X, right? So every drug or device has a treatment effect, and you can see here in 3 different populations of patients, overall the drug is working. The median, which is kind of how we look at overall response rates, there's a very significant sizable effect. However, you can see a lot of patients fall outside of the treatment effect window and may not be observing the same safety and efficacy as the mean. So this really doesn't translate well unless our clinical trials are really expansive. The problem is clinical trials are really expensive, so there's a big challenge in the US. How do we develop this personalized, individualized data for drugs and devices without having to spend $28,000 per patient to generate that data? And so as I highlighted, the phase 4 trials really are focusing on subpopulations. In liver disease, typically the most complicated patient, the highest risk of a drug to fail or develop safety effects are cirrhotic patients, those with advanced fibrosis in the liver. Therefore, they're typically minimized in trials to get the drug approved, but then that's the population we're putting it into, and so that's an example. There's lots of reasons why these phase 4 studies don't do well. They're really expensive. They're underpowered. It's a bargaining process with the FDA. Okay, 100 patients, African Americans, and all right, but without really confidence intervals around the data you're going to produce. And we all deal in confidence intervals, and I'll show you. As clinicians, you may not realize it, but we have to understand what's the confidence in the data that we're basing our decision. And if it's this big, our decision is probably a flip of a coin. So the goal of phase 4 and other studies are to narrow confidence intervals, certainly less than 5%, and in theory, much closer. And often these studies are not high priority for pharma, and it takes years to enroll, and by the time the data comes out, they're obsolete. So we've been thinking a lot about this, and those of us who have watched and been part of this Hep C evolution kind of knew what was coming. But so real-world evidence has a potential role. And there are different types of real-world evidence I've just listed. So observational registries, there can be interventional registries, and then kind of randomized pragmatic trials, which are a kind of the blend of a clinical trial with real-world observational data. Now, the EU actually has already thought about this. And the EU, this slide just shows you that the EMA, their regulatory agency, when they approve drugs based on the risk of the category, they will often require that a national registry be established. And so you can see it depends, in the exceptional circumstances, they're trying to get a drug to market really quickly because there's a massive unmet need and lives can be saved. You probably don't make pharma do all the extensive phase 3 studies. You get the drug in place, but you then have to put in a safety net to make sure you're collecting data so you can either alter or adjust your decision. So we don't have a system like that in the United States. But for those of you who have followed the political legislation, the 21st Century Cures Act, which is now law, basically suggested to the FDA that by 2021, they needed to come up with real-world evidence, guidance, that can be used effectively to advance drug development and drug safety and efficacy, probably even going beyond what the EU has done. And so the real-world evidence I highlighted here, it requires the FDA to evaluate the use of real-world evidence to help support both the approval of a new indication and also help support and satisfy post-commitment approvals. So the idea is, instead of company X going out and doing 100 African Americans to try to broaden the knowledge around that, maybe company X sponsors a 12,000 patient cohort where we have a disease cohort and learn a lot about how that drug and others come in and perform. So I think that was where this field is quickly evolving. So if you're going to suggest, let's get real-world data, what does that mean? And everyone has a different definition, and this just shows you there's lots of places to go, right? Claims data has been the classic one, but claims data isn't linked to what you and I do every day, which is see patients with findings and adverse events, et cetera. But you can see the EMR data. There are surveillance, morbidity, mortality, state vitals, cancer registry, all kinds of real-world data that's being collected anyways. And so these real-world evidence cohorts, once they're formed, become actually really important. It really helps define natural history. So I know Dr. Levy's involved in one with PBC, others involved in one with cirrhosis, Hep C. So you can actually define the natural history of the disease in your population, and then really help find the gaps in where they exist that directs either interventions or, for, say, pharma, the pre-discovery, drug discovery pathway. So a lot of real-world evidence actually feeds the pipe to say where are the gaps, where are the needs, and allow us to think about even clinical practice patterns and where there's opportunities, say, for implementation science grants. However, kind of what I'm going to focus on is where we've taken this is trying to use real-world evidence to get drugs into the market quicker, and then once they're there, to understand how to use the drugs better. And so that has kind of been the easy picking, because there's a lot of interest at the FDA and there's a lot of resources if somebody can do it well and provide an advantage. So HCV target, it was really created, I think back in 2011, with at just about the time a number of us were sitting on advisory boards or involved in phase 1 studies and early 2, and we knew there was an explosion coming of new medications. A number had been taken off the market before they got there because of adverse events, and we knew it was going to be kind of the Wild West. And so we kind of thought through that we need some organization that can maybe help develop an infrastructure that how these drugs could be studied. So we formed HCV Target. It was really to improve populations not represented or underrepresented in phase 3 trials. And to see, we all do as clinicians, we do multiple experiments every day. And if you can capture those experiments and statistically evaluate those, you often don't have to run clinical trials on, is this drug better at 8 or 12 or 24 weeks? Because if we have tens of thousands of patients, docs are doing that in similar patients, and you will learn an outcome right there. So that's the idea of using clinical practice to help evaluate these regimens, especially when docs go off-label. We learn a lot of information, and sometimes you're going to see that an off-label drug led to, just by collecting real-world data, led to a new label from the FDA. So there's a lot of, I think, value to patient and clinician community. The reason most of these are set up, in theory, is mainly around watching for adverse events and trying to understand how these drugs' safety profiles look like in the real world, especially under real-world conditions where the timing of the drug delivery, the dosing is not always consistent with clinical trials. And then, like most registries or cohorts, we like to do some science, and we've been collecting a lot of data. The difference here is we went to the NIH and said, we'd like to do a 10,000 patient cohort for Hep C and follow them for 5 years, but not define a specific drug. We want to define the disease itself and see how these drugs come in and out, and you can follow longitudinally people who failed. And the NIH said that's going to cost $100 million. We don't have that kind of money, and especially NIDDK, which is funding these. So then we started talking to pharma, and we went to pharma with the model that as you're in phase 2 or 3, again, contribute X dollars and give us 2,000 patients worth of 5-year data. And so that model worked really well. Those pharma agreed to allow their data to be publicly displayed. So it's a disease cohort, knowing their drugs would come in and out of it, and they would get the benefit also of just seeing what's happening in the market, but more seeing how drugs are used and how their drug is used. And the only reason this is a big win for them is only if the FDA says by doing this, you just got out of $100 million worth of post-commitment approval studies. And we have to prove benefit. And so I'm going to try to then take the story as to kind of where we took it from there. So obviously we were successful, or I wouldn't be here today. So this was led by academia, supported by pharma initially, and some of the federal agencies came in, brought in patients at the very beginning, and then really developed, I think, the first true paradigm with the FDA, and it started in 2012, I'll show you, but eventually ended up with an FDA-wide MOU to allow companies to use registries for observational or for PMR PMCs. And I'll show you a little bit about this. Again, this is set up very much like an NIH model. You'll see it in a second. Our goal was to improve patient care, to publish, get tenure promotion, all the same things that we all chase. And this was a unique research opportunity to develop a kind of an innovative strategy for doing clinical trials. So again, the Cures Act came out and is going to mandate the FDA do something by 21. So we were fortunate that we had some trust with the FDA, and we started a very non-threatening memorandum of understanding at the University of Florida with the FDA, and it basically just said, look, so the FDA actually has a hard time talking to clinicians. They love just to sit down and talk to people who have expertise in the disease. Typically what they hear from are the pharma folks or people on advisory committees. And there is really not an obvious pathway where you walk in like to talk to your congressman and say, hey, I got a great idea, or I think there's a trouble here. Anyway, it's an interesting, and so the FDA, at least from our impression then, was they're starving for academic collaboration. And so we set up a collaborative agreement where we would develop collaborations, joint meetings, which eventually transformed into co-developing a data set and developing a model for moving drugs more safely and quickly through pipeline, at least in Hep C, and that model is now moved to other disease areas. So here's the structure. It would look like an NIHU grant. A good friend of mine, Mike Freed, who's actually president of the ASLD this year, sits at UNC, and they do one thing really well. They know how to run data coordinating centers. So we said, all right, we're going to separate church and state. We're going to have the clinical coordinating center, in this case in Gainesville, data up in North Carolina. But the most important part is the steering committee. Oops. That's usually put like a red piece of tape, says do not touch over that button. So we have a steering committee, which are all the academic PIs from across the country. Industry gets to sit in on all of the data discussions and presentations. The FDA has participation fairly significantly in an advisory capacity. We have a massive patient organization that helps with this, and then again, currency of what we all want to do is disseminate, and to disseminate you need to publish. So often these registries do a lot of surveillance without getting data out. And I'll show you so this is a very, it's a lot of effort to keep publications coming and abstracts and anyway, and we do, I think, fairly novel methods for distance QA and QC, and we've set it up. I'll just cut to the chase. So you typically would cost us a little bit under $1,000 to follow a patient and get all of their outcomes for 5 years. And so that's getting down. It's not where it needs to be. You can see what Corey's trying to do now, maybe get it down to the 200 to 400 range. But 200 to 400 range doesn't pay for our staff's inability. That's why we're moving much towards a patient-centered self-recruitment and self-reporting. So this is the model. We were able to gather most of the big academic centers in the U.S. This started out as a U.S.-specific effort, but interestingly, as pharma develops drugs, they almost always have a European component. And in fact, sometimes there's a European predominance of patients entered. And so it was a request that we responded to from the FDA to say, we would actually, can you guys grab European patients to participate? There were some legal challenges that didn't turn out to be big ones. So anyway, we have about 10 or 15% of the patients in Target coming from Europe. That also helps the European regulatory agency act on findings from the U.S., because you need some representative sample. So anyway, we didn't start out thinking that. It became a value added from the FDA, obviously pharma as well. So again, this is kind of a give and take to try to find what's the right opportunity. But clearly, these are the major academic centers, and Miami has been a close partner throughout, actually from day one. So I want to dispel the notion that this is just a registry, because a registry, can't remember if you guys have Cerner or Epic, you can set up a registry that just follows patients. This is a very active process. Describe it more as an observational cohort. It's kind of led by hypothesis generating kind of academic thought, and then a lot of the data analysis or data populations are occurring related to areas of interest. So what we do is we grab University of Miami, the entire electronic health record data from 55 universities across the country and 6 in Europe that are participating. That's redacted, so, and then at least at a number of them, we set up an electronic, it's called Red Eye, it's an open source, where all of the labs, medications, things like that are flowing electronically to get away from data abstraction. So we're starting to mix a little bit of innovation as opposed to, but we bring all the EMR data into the data center, and then it's hand entered in terms of key clinical endpoints. So there's still that component that we eventually like to get rid of. But people are trained on data standards. So instead of each different site interpreting an adverse event or an outcome differently, there's a standardized data dictionary, so everything is brought in under the same analytic strategy. And then there's a dissemination plan. So the thing that also I think for those of you who, and again, this can be done in any disease. It's usually led by clinicians who are passionate and influential in the disease and have an opportunity to bring in hypothesis generating research into a cohort. But to do it, in order to fully recognize the advantage of the data you collect, there's a couple of things you have to do. The FDA nor pharma can take the data and ever use it other than, oh, I see a publication, unless it's 21 CFR Part 11 compliant. That's a fancy term for a separate type of data set that has to use what's called CDIS standards for how the data linkages are established. It's actually not that hard. REDCap, which is all over this university, has been deemed by the FDA last year as a 2111 compliant, and it just need then the statisticians to help work through CDIS compliance, which is that you can monitor and track every data entry, you know who touched what, and so anyway, if you're going to put together a data set, these are small things that make the data extremely valuable, because then it can be used to update drug labels and can be used by pharma to satisfy commitments back to the FDA. So that was actually a partnership with the FDA to help develop the data sets. We also standardize every medication, right? How many different ways are there to describe a beta blocker, an ACE inhibitor? Some are diuretics, some are antihypertensive, some are both. And so we've been able to use WHO drug and Medra to have basically FDA-defined standards for what our medications are and assessing adverse events. So again, we're putting what typically goes into any data set into terms that the FDA can use and can then compare to phase 3 data sets. We also do a lot of monitoring. Some are 100%, right? So it's probably the same in every university. Medicare fraud billing is key, right? So we monitor every single bill that leaves the University of Florida for Medicare, but maybe for a commercial payer, we don't, and we only do risk-based billing. And so this is the same kind of thing. What are your absolute, not in this case, fines? What's your most important outcomes? In this case, it was safety, and did Hep C patients get cured? So we would physically verify 100% that a virologic, say, outcome was there, and certain safety signals, which had already been agreed upon by a steering committee and the FDA, had to be tracked down to the nth degree. And so far, we've actually done pretty good. When you get the entire EHR, you have fairly good low missing rates. Now this will change. So most of these Hep C patients are committed. They're going to keep coming to the University of Miami because they got treated, they then need cancer screening, and they stay within a health system. Where this falls apart is when they start to leave that health system, and then you lose that EHR connection. At the very end of the talk, I'll talk about an innovative strategy where Florida, we are now linking all the health systems to try to overcome that. But because at least the Hep C patients historically have been very dedicated to staying in their clinics for follow-up, we've really had minimal loss of data, and less than 5% loss to follow-up is really a pretty good number for this world. So this is just a hypothetical, the type of data that we would develop for these cohorts or has been developed. There's baseline characteristics, but the most important thing are the longitudinal nature of events, when drugs come in, when adverse events come in, when comorbidities come in. It's often very hard unless you have that longitudinal data to understand, was an adverse event related to the beginning of a medication? What was the drug-drug interaction that came in from this ConMed? So again, the data set was developed kind of almost the way an FDA evaluation would look at when they come to evaluate a phase 3 data set. And we try to get all the biopsy data and all that. So far, we haven't been able to visually bring in imaging, and there's a project underway. Obviously, we bring in the finding, and so we have a pretty rich data set. Okay, so I'll start with the first story. Back in 2011, the first new oral pill for Hep C came out, and it was combined with the old horrible drugs, pegrabavar and PR. And what you'll see is just some of the early phase 3 data that said, hey, cure rates almost double. No doubt that drug's going to get approved. Serious adverse events looks okay. Discontinuations due to adverse events, which I think is probably the most important signal. If people are getting adverse events, that's one thing. If they're actually stopping their treatment because of it, yeah, there's a little signal. It didn't look too bad. People with cirrhosis, very small numbers, 21 patients led to this drug's approval. And back then, this was where we knew the drug was going to go, almost 100%. Because someone with mild liver damage was probably not going to continue. They knew it was coming in 5 years. And so I think, again, here's an example where you'll see in a second what the world found is that anemia, which was significant safety signal, and blood transfusions looked like they would be manageable. So the drug hits the real world. And actually, this is a French registry that had been set up to evaluate this in advance of the drug's approval. And what you see is a very different profile when the drug hits the real world. Cure rates, it turned out, were actually still pretty high, but adverse events basically doubled, discontinuations doubled, and the over 50% of people needed erythropoietin to get through the drug regimen, and 16% were being transfused. So, that's the value. I think we all felt it immediately in the clinics, like there's something going on here, but to get together a large data set really immediately led to the FDA to step in, ask this company to actually start putting in new monitoring guidelines, then immediately responded. So it was a good example of real-world evidence immediately allowing clinicians to take safer care of patients. Actually, this company, Vertex, was the first company who invested in this target model because of this. They knew they had a big issue and they needed to better understand it. And so they helped us follow thousands of patients to find out what was happening to them. And more importantly, what strategies? Did you reduce drug? Do you throw in erythropoietin instead? Is it better to give a blood transfusion? Those would all be multiple clinical trials that would take decades to figure out the right answer. And so the goal is in these big data sets, clinicians make those experiments on their own, and you learn from what's happening in clinical care. And so that actually occurred. It turned out that people reduced ribavirin. We used EPO early and often, and blood transfusions in the U.S. were often not done except for very sick patients. But here's a great example of a drug that was tested in patients with mild disease. The clinical community's need was in advanced disease, and the drug basically blew up when it hit the market. And so having the warning sign in the clinician's office is one thing. Having the safety net to collect the data, understand it, and then change practice, I think, is the real utility of some of these cohorts. So another story, again, this is a little heavy for people who weren't in the Hep C world, but so the first 2 oral pills for Hep C were both going to be approved. These were the second generation of the one that blew up in that other study. They were both going to be approved with standard interferon ribavirin, so horrible side effects, trying to get away from that. So I'll never forget, we were at the FDA because we would meet regularly, and before this drug got launched, because we were setting up to make sure we captured this, and we said to the head of DDA, Deborah Bernkrantz, said, I think clinicians are going to take these 2 drugs, order them separately, and then put them together, because this will all of a sudden be the first all-oral therapy. And they're like, we don't really think so. So what happened is the company, around the same time the drug was launched, had a phase 2 study showing that these 2 drugs had pretty good outcomes. So clinicians armed with a little bit of knowledge and a huge need, because interferon is probably eliminates 60, 70, 80% of patients who would potentially be candidates. And it really kind of changed the paradigm, because physicians had knowledge that these 2 drugs off-label could work, and it's up to the payers now, but if the payers allow it, clinicians were going to experiment because people were dying, and these are sick people who couldn't wait 3 or 5 years. So quickly, in 2014, when Cimeprevir was approved, it became the #1 prescribed regimen together in the United States. In fact, the company made $4 billion off an unapproved indication. It just shows you the power of new therapies and when they fill a gap, what happens. So I'll just show you again where Target came in. So we've been tracking this, and so in January 1st, the drug had been approved with interferon, and within a few months we were able to follow almost 650 people who were getting off-label combination therapy. The company actually started a phase 3 trial months after, but given the phase 2 data, asked the FDA to approve this this drug, change the label to allow it to be combined medically with this other drug. It's a positive story. The company, or the FDA, asked the company to ask Target to report the data back to the FDA. The data was very positive in terms of outcomes, and well before the phase 3 trial was even close to completion, well before anything was published, well before anything was vetted at the FDA advisory committee, the FDA granted a label expansion based on a small phase 2 data set and based on a big real-world data set. And to me, that sets the model for how really critical drugs that need to go forward to meet a big gap in medical care companies are going to start using real-world evidence to say, we will get good phase 2 data. We'll set up a massive study to follow those patients who are going to be off-label. And then, oh, by the way, we'll do a phase 3 study. So this just shows you the phase 3 trial was about a third of the number of the patients that were just followed in the observational cohort. And then we had published a paper just to say, hey, how close did the real-world evidence prediction in a much sicker population do with the eventual phase 3 trials? And this just looks at cure rates on the vertical axis. And it says, you know what, the real-world data was a little bit lower, but that's good because it was in a sicker population. But overall, it can't take the place of phase 3 trials because these are not randomized. There's biases that come in when physicians make choices. But again, I think the first example that I'm aware of where real-world data allowed a drug to be approved before a phase 3 study. And I think it's one that will be copied multiple times. Okay, so now we move into the new world where we have every drug from every company. And for those of you who don't know what SVR12 is, that's someone gets treated and cured. So, but I want to highlight a couple of things. One is what happened in this drug development and what's also happening in other drug development, which there's a big criticism, is there has never been a head-to-head trial in the hepatitis C world with all oral therapies. So the FDA has allowed companies to set a target range of what they want to see in terms of an outcome, and they've allowed them to do single drug studies. So from a clinician and a patient, and especially a payer, we don't really know, and we never will know, which drug is the right drug for this person in this setting. But it's really expensive, right, for the first drug. So you understand the rationale. $100,000, right? This was the most expensive pill ever produced when Gilead first launched their first drug. And to do that study, to compare your drug to Gilead's drug, you would have to buy, billions of dollars of drug to power up a study. So the FDA could actually probably force that not to occur and to have to provide drug-free for head-to-head. But anyway, so that's the state of where we were at. So we're also very interested in how do we start to compare these drugs, which is challenging when so many come out that appear to do so well. So the other thing, again, even though it looks like everyone is being cured, because that's what phase 3 trials do, they take highly likely to be cured patients. I'll just focus on one of the drugs, ledipasvir, sofosbuvir, and just show you that the entire experience in all the clinical trials, there were 104 blacks, in a naive population, and there were a limited number of cirrhotics. And so it leaves a lack of confidence in knowing how a patient in those subgroups is going to respond. And this just kind of shows you an analysis of the phase 3 trials of where are the evidence gaps. And what you'll see is age, race, and these are really big confidence intervals, right? It means that you might have a 70% chance of difference in response, and especially cirrhosis, if you have it. These are big confidence intervals. Usually clinicians want confidence intervals, I would say, under 5%. And for a lot of decisions, even in 2% or low. So that's just how much confidence do you have the result is going to apply to your patient. So a lot of what we've done in Target is to try to reduce confidence intervals. You can't really do head-to-head comparative studies. I will show you that we've evolved to that, but at least we can give clinicians a better confidence in that population. What does a 65-year-old African-American cirrhotic do as opposed to what has typically not been available in clinical trials. And so without going through the exact details, you'll see the same regimen with the same phase 3 study that you just saw earlier now reporting out 2,200 people who got this regimen. And I just wanted to pay attention to the confidence intervals, which are overall, right, inside of 1%. But even in, where's the cirrhotic group, talking about confidence intervals that are only moving by about 3.5%. So you'll also see that, yes, cirrhotics get cured at a lower rate, 93% versus the general public or non-cirrhotics. But you'll see we still have pretty good confidence that they're going to have a high cure rate. And so we do that across all of the populations that we report on. So here, again, it's nothing more than the power of large sample sizes to gain, in this case, confidence around efficacy. The same is done around safety. So another thing that came out early on is the first drug that got approved with an 8-week label instead of a 12. And that at $100,000 a pop, there would be a great desire to use 8 weeks. It turned out that 8-week label was only given to a subset of patients who had low levels of virus, because those with higher levels of virus had higher rates of failure. Now, overall, the entire difference between 12 and 8 weeks was only about 3%. And so most times they would have gotten an approval, just treat everyone for the lower amount. But in this case, the FDA didn't look at the efficacy outcome of cure. They looked at, with small numbers, relapse rates. And what you see is that relapse rates went from 2 to 10% in a at-risk population, those with high viral load. So in a small number of patients, the FDA said, you know what, there's probably enough societal benefit that we should allow an 8-week regimen because the cost difference would be enormous and we don't think there's a big risk. So then the world had to try to decide, is this the right thing? So again, this slide is really the best example of real-world experiments. So within HCV Target, we have tens of thousands of people being treated, and we looked at a cohort of people who would have qualified if they were in a clinical trial for 8 weeks of therapy. And just because some docs believed the data and some didn't, they got either 8 or 12 weeks. And again, in a much larger subset, if the company had to do this trial, it would have taken 4 years. It would have cost, $60 million. And within about a year, we were able to generate data that at least clinicians felt comfortable, we felt comfortable to say, you know what, there's a minimal, failure rate difference, but overall, statistically, it doesn't look like if you use the criteria the FDA suggested, you're going to fail. And so the same thing was duplicated by other real-world data sets. So again, one of the nice things about real-world data sets is they can confirm or not confirm some hypothesis-generating modeling that the FDA often does. And so the FDA is much more likely to model a more attractive drug regimen if they know there's something that's going to be able to evaluate that going forward. The other thing we found that, again, this is just big data and analyzing big data. We actually looked at our data set and the statisticians kept saying, look, there's something about using proton pump inhibitors. Now, one of the drugs in this combination requires an acidic environment to be absorbed, so there's a theoretical basis. The phase 3 trials were not heavily regulated around PPI use. And so through a lot of fancy statistics that I pray no one asks me about called inverse probability weighting, we were able to pretty conclusively show that even though 97 versus 94% doesn't seem like a huge number, it's very significant, and the association with proton pump inhibitors was there. So the FDA took this information and adjusted the drug label, sent out Dear Doctors letters to say, look, because this is a modifiable risk, right? All you have to do is ask your patient to take his PPI in a different rotation or switch to a different antacid strategy. And so, again, here's an example. It's not truly a safety signal, but it's a drug-drug interaction that was not appreciated in phase 3 trials that was picked up in the real world that actually costs nothing to actually improve cure rates in those people. And just another big data set came from another group, and in this case, they actually took it the next step. They said it's not just the PPI itself, it's how much PPI, so it's how much acid suppression. So that's just a bunch of examples to, I think, hit home that these cohorts have utility, but they have utility if there's an academic component that I think is driving them as opposed to companies setting up registries. Because you get to ask questions. That PPI data would have never hit the light of day, I think, if it had been sitting inside of a pharma registry. So there's a real benefit for academia having final control of what data gets disseminated and how. So the other thing, since we built this kind of cool network, I won't go through it, but we've been able to do the first head-to-head study ever now because we have a network and we're tackling a big one that's going to be presented in Europe in a couple of weeks. We put a bunch of other PCORI grants in, and we did something I think that's following the oncology model, is we went to company in the middle of a phase 3 program and said, would you basically take a phase 4 study and build it into the phase 3B and let us run it? In other words, you give us drug, we'll put it in the real world, and it will be one of your phase 3B studies. This was AbbVie, and they're not used to being anything other than conservative. Anyway, in discussions with the FDA, they built their phase 3B study into Target. And so these phase 3B pragmatic trials are really, I think, important because you can get drug, put it in the population, and then the healthcare delivery delivers the outcomes. So it's very much like the cancer trials. If there's an unmet need, even before the drug's approved, we can, for that indication, we can put it in that unmet need, and then the healthcare system generates the data. And if you can collect that in a fairly rigorous fashion, you can do an actual clinical trial inside it, and these are the pragmatic trials. They're much better because they're delivered the way we deliver real-world drugs and real-world follow-up and patient adherence and compliance are not strictly monitored. So they have a lot of advantages. Also, the threat to why it's not as a tightly controlled population. So for people who fail these new oral pills, there's very few of them. But the ones that do, there's only been one potential drug to treat it. This GP combination drug just got approved, but it had never been studied in this failure population to any extent. And so we were able to convince the FDA and the company, let us take a crack at it, because there are not many failures. It's kind of like a rare disease. So, but we have a list that, hey, we have 500 people who failed these drugs that actually look like they fit the definition of what we want to put in a clinical trial. So we said, you give us the drug, we can perform this study and deliver it kind of in time for even your phase 3 submission. And we tested a couple of different concepts. Can you go shorter up top in patients with minimal disease, or can you shorten therapy? Because the standard therapy, at the time, we didn't know what the drug label was going to be. This was a little bit of a guess, just clinically how we think the drugs would be used. And so we put a few variations. So I just want to show you the data. This is on the left, 12 weeks and 16 weeks for patients without advanced liver disease. And then this is the hardest patient. These are cirrhotics. And can we shorten therapy by using that old horrible drug Ribavirin, or do you have to go 16 weeks? We actually stopped this arm early because there were 3 breakthroughs, which is a really rare event, which means trouble. And so that was a big conversation with the company and the FDA in the end, since in this case I held the IND, we had the final say, and we shut down that arm. In the end, it was the right thing to do for reasons that we'll probably highlight in the second slide. So it looks like when you look at it in a different way, 16 weeks is going to be the label the FDA eventually gave this drug. And when you look at how the drug performed, regardless of what else was in it, 16 weeks appeared to be more ideal because even a 10% failure rate is unacceptable in this population because they develop severe resistance to the drug. And then there are certain populations, subgenotypes, that actually the drug is perfect for. So it's just a really unique example. Again, one of the first examples, I think, where an independent cohort organization was allowed to do a phase 3B trial, and this will enter both the US and European labels. Again, this was not a company's run study. This was a study run and handing over. So again, it brings a lot of credibility and I think acceptance to the data because it's basically run by academic pragmatic docs. So we've done a lot to disseminate. We've got one of the things we've really strived to do is to keep people motivated, is to include a lot of people as authors. this data, I've just highlighted a couple of the data sets. This data has been used by 7 companies, FDA, CDC. It's been incorporated into the US, European, and worldwide guidelines to help teach future people how best to use these drugs. And then this past year, we had a NICE award. It's a very rare Regulatory Science of Excellence award handed out by the FDA. Because, again, I think they believe this is a value-added proposition for going forward. So I'm going to end on something that I think is a huge opportunity. So, again, I didn't know anything about data, cohorts, and then a weird thing happened 10 years ago, and I switched jobs, and all of a sudden I was around people who do big data and do outcomes research, and so, the idea of what to do came from that. So, but building the Hep C model was okay, but it has limitations because it still requires people to pull down certain data sets. So we started thinking about, it's on a timer. Anyway, we started thinking about what would it look like to bring together all the health records in Florida. And so UF and FSU and the University of Miami were the 3 big academic partners to say we're in. And then you see a number of other health systems in the state have partnered, and then we've got a lot of other partners who are interested. And so what does it mean? And you guys have this available now. We're trying to promote it here at UM. So right now, there are 15 million lives represented in what's called this One Florida Data Trust. And you'll see it's loaded with longitudinal data. And there is no reason we can't be building cohorts in every disease throughout the state. This is really rich data. It's a little complicated. There are honest brokers. At Gainesville, I can't see your patient. I can't contact them, but you can. And we can either use it for cohort discovery, but also what we're trying to do is use it for outcomes. If we're going to deliver a drug or an intervention or do pragmatic trials, we can use these data sets. So this is a huge collaboration so far. It's supported. Unfortunately, we've had to just pay for it grant by grant. Luckily, there's a lot of grants that are using this mechanism. We've had some recent discussions with the legislature about potentially this is a huge opportunity for the state, and I think we might see some activity. But just more importantly, the CTSI here is starting to bring this on board, and there's also the ability, I should have brought the slide, if you're interested in a cohort discovery, say, I want to know how many African American women over the age of 60 also have diabetes and renal insufficiency. You can, on the same screen, say how many are at the University of Miami, how many are at One Florida, and how many sit in what's called the NCATS consortium. So you can go from Miami, 1 million, you can go to One Florida, 15 million, and then you can go nationally to about 200 million. And so every grant should have that kind of analysis just around cohorts. So that is at the tip of your finger. So it's a really important resource that I obviously am excited about. We spent a lot of time and effort building this. So I want to give you one example to leave you with that we're working on between UF and Miami. So obviously Florida has a lot of leading causes of death. Cancer and liver disease are 2 of the top 10. It costs a lot of money. And there's a lot of interest in this real-world evidence. So this one slide kind of summarizes everything we've done. We're really interested in cancer screening. And people with advanced liver disease called cirrhosis are at risk for cancer. And there are simple guidelines that say if you're at risk for cancer, you should get a very cheap ultrasound done every 6 months. So we wanted to just ask the simple question in the state of Florida, how many people with known cirrhosis are being screened? And so I won't go through all of the ways we narrowed down a very pure cohort. It actually had to have claims data in case it switched healthcare systems. But we had about 11,000 people who we know have cirrhosis. And then we were able to look at the screening guidelines. And what's shown here is only 4%, and this includes people coming out of the Miami area, coming out of the Gainesville area, coming out of the Mayo-Jacksonville area, but also includes much of the state. Only 4% of people got what we would consider appropriate screening. 17 got some screening, but it was not the recommended screening. And the bottom line is 80% of people got no imaging at all after their known diagnosis of cirrhosis at any point going forward. So, huge problem, but a huge opportunity, right, for the academic world to come in and develop interventions and strategies. We thought we would see more health disparities. What we saw is obvious. If you have insurance, you're more likely to be screened. If you see a GI doc, you were much more likely to be screened. Interestingly, there was no difference in some of the classic racial disparities. And we did see people who were sicker often got screened more, but that's probably because they were more likely to be in the healthcare system. So again, here's just a simple example of ask the question, hey, I wonder if people are following the guidelines. You know, this doesn't take a lot of time or effort. And then, you know, we want to write a bunch of grants to try to figure out how we can improve this kind of more healthcare delivery component. So I'll end up with this. Screening for liver cancer using these data sets was horrible. I think I just kind of highlighted at least the general conclusions. And so I'm just going to end. Hopefully I've convinced you that real-world evidence provides really important information, both natural history as well as effectiveness of new treatments. It's contributed to the optimal use. It allows us to use drugs more safely in the populations that we're treating who are not phase 3 trials. And I think the real message is the regulatory agencies have embraced it in the past at a small level, and they have to embrace it by law in the next few years. So for all of you clinicians who have interesting ideas, I think, you know, we should be all talking about building some of these cohorts for Florida, which is, you know, a really unique, you know, the Florida of today is the nation of tomorrow, right? It's older, more diverse. It is the population that we will be as a nation. So with that, I want to thank Jean for the invitation. This is an honor to come for the Leon Schiff Memorial Lecture, and just want to thank UM for the invitation, and enjoyed my stay here so far. Thank you.
Thank you, Dr. Nelson, for introducing us to this interesting paradigm of how the government and academia can work together. Individuals that have questions, please come up to the front and ask them. I too want to thank the Schiff family for sponsoring today's lecture and the lunch, which is an unusual and appreciated addition. I want to caution everybody that, not caution, but to tell everybody that next week is the annual Department of Medicine Research Day, and we'll be having grand rounds on the 7th floor of the Lois Pope building at the same time. Thank you all, and I hope to see you all there. So, one Florida,
Yeah, actually, Alvin just texted me on NBC. Sorry, I'm going to miss the right. Alvin's a good investor.
So I wonder about the